Okay, Bree, where are you? Okay, so yeah. Okay. Okay, so there's the room number that you put in for Socrative. Where'd you go? Okay, some of you are logged in, I see. Okay, 17 of you. And a lot of you are finished, I see. And we've got a few that haven't finished. I wish the online tests that you do in Blackboard were this way. You could see how people are doing. It should be your best score that's recorded. That's the setting that I put in. And if it's not doing that, then something's wrong. That's the score that's supposed to be entered in Blackboard as your top score. Well, none that you can see. Some people can see their scores, some people can't. And I've tried to figure that out, and McGraw-Hill can't figure it out either. So, all right, so let's go to export, download. Okay, so that's the, oh boy. Okay, so let's go back. How about we just go back to the quiz? Let's do that. Okay, let's go through this quick. Glycolysis is used by cells to degrade. It's used mainly to degrade glucose. You can use it to degrade amino acids, but it's not used to degrade all amino acids. So that's not the right answer. Plenty of amino acids feed into glycolysis, not all of them. The other group of amino acids feed into the Krebs cycle. So all amino acids, that's not correct. Lipids, no. Lipids are degraded by beta oxidation. So that's not right either. Ribose, no. You don't use glycolysis to degrade ribose. You use other means to degrade ribose. Glucose and other hexose sugars, that's the main function of glycolysis. So B is the right answer here. Krebs cycle is both a catabolic and anabolic pathway. That's true. Some of the intermediates, the metabolites in Krebs cycle are used to make very important molecules. So for example, several Krebs cycle intermediates are used to make several amino acids. Just a few examples. So oxaloacetate is used to make aspartate. The amino acid aspartate, and from aspartate, you also make another amino acid, asparagine. Then there's another Krebs cycle intermediate called alpha ketoglutarate. That's used to make glutamate. And it's also used to make glutamine. Glutamate is also used to make a couple of other amino acids like proline. Now, I don't require you to remember this. All I want you to know is that from the Krebs cycle intermediates, you withdraw metabolites and you use them to make various molecules. Another molecule that's very important for biosynthesis that's made in the Krebs cycle is this molecule, succinyl coenzyme A. Succinyl coenzyme A is withdrawn and is used to make heme. What's heme? Where have you seen the word heme before? Right, hemoglobin. Heme is the planar molecule that's attached non-covalently to the hemoglobin polypeptide. That's the part that holds the iron that binds the oxygen. Heme is used for hemoglobin, it's used for myoglobin, it's used for cytochromes. It's really important. And this is made from a Krebs cycle intermediate. So because of that, the Krebs cycle is catabolic, but it's also anabolic, so it's so-called amphibolic. It's a little bit too versed in what it does. Which of the following pathways is not involved in degrading amino acids? Glycolysis, yes, it is. Krebs cycle, yes, it is. Beta oxidation, yes. Branch chain amino acids, branch chain amino acids. How many of you athletes have seen branch chain amino acid supplements? Comes as a white powder. BCAAs, they're sometimes called. Hannah, you've seen them? Anyone else? You've did. We actually tested them here. We tested endurance athletes to see if they would extend endurance. They don't. We also tried power to see if they increase power. They don't. We did, however, show, and other people have shown, they do help with recovery. So if you take BCAAs, don't expect to get a bigger workout from them. Do expect to recover faster from them. BCAAs are degraded by beta oxidation. When you get to the final carbon skeleton, you degrade that via beta oxidation. The one pathway where you don't degrade amino acids is the pentose phosphate pathway. You don't use that for amino acids. So D is the answer there. What molecules are made by the pentose phosphate pathway? We talked about this at the end of Friday. What do they do? Pentose phosphate pathway, what does it make? It makes 5 carbon sugars for nucleotide synthesis. Ribose 5-phosphate, that's made from the pentose phosphate pathway. You don't make hexose sugars from the pentose phosphate pathway. You don't make glycerol. No. And it's essential for the synthesis of nucleotides, DNA, RNA, things like that. So C. And then 5, all fermentation products culminate in the production of ethanol. If you miss this one, I should bump you on the head with a Spätzle. The answer is false. False. Do you guys know what a Spätzle is? It's a German noodle. In the south of Germany, it's a type of noodle. It's very tasty, I understand, but it's a noodle. So 100 lashes with a wet noodle. Hit you with this. Okay, it's early. Never mind. Yeah, false. That one's false. You make organic acids. You make lots of interesting other alcohols that are not ethanol. You make plenty of other things. Yes, you make some ethanol, and some fermentations, in fact, make mainly ethanol. But all fermentation pathways do not culminate in the production of ethanol. No. All right, so go through and think about those again if you missed some, and we will hopefully get you going. Oh, Bree's here. Good, Bree. Hey, Bree, you feeling better?
Yeah.
Cool. So did you test positive at all?
No.
So will you be back to us so shortly?
Yeah, I'll be back. So I won't be there on Tuesday because I'm in quarantine until Tuesday at 8 p.m. So could I go to Wednesday's lab?
Let me think. Wednesday, folks, what do you think? Should we let Bree come? Yeah, they're nodding their heads. So yeah, you can come Wednesday.
Okay.
Mrs. Schaefer has your lab book.
Yeah, I've emailed her.
Okay, so, all right, excellent. Okay, then let's take a look at what we're meant to study today. Today, we're going to study the genetics of bacteria, and I have a little handout for those of you I haven't given this to yet, which is most of you. Where's Natalie? Did Natalie tell me she wasn't going to be here today? And I forgot. Sorry? Okay, I wonder if she told me she wasn't going to be here and I forgot because. Okay. So I was gone all weekend. I was in Florida this weekend for a wedding. When people ask me how'd the wedding go, the answer is always the same. Interesting. This friend of my daughter, she got married standing in the ocean. Oh, you already got one. So there. Oh, I got one too. So, okay. Did everybody get one? Did I forget anybody? All right. And this is my visual prop for the day. So this will be the subject of the day, DNA. Give a little whirl so you can see how it goes. So this molecule stores genetic information. When we say genetic, what do we mean? We mean characteristics that are inherited from one organism to the progeny. From the parent organism to the progeny, if they're inherited, they're genetic. Inherited things are not things that you learn. For example, many songbirds learn the songs from their parents. Is that genetic? Well, it's passed down from progeny, but if those birds are raised by different parents of different species, they'll learn a different mating song. So strictly speaking, that's not generic. That's what's called cultural inheritance. So it's not really genetic. For something to be genetic, it has to be inherited by virtue of your descendancy. So that's what we mean by something that's genetic. So genetics is the study of inheritance. It's the study of how traits are inherited and transmitted from parents to offspring, how those traits happen to be manifested in an organism and then in that organism's progeny. It's also the study of the structure of the molecule that stores genetic information, that would be DNA, and then how that material is accessed through expression, through things like transcription and translation and what have you, and then how that material changes due to mutations. When we talk about all the DNA inside an organism and the sum total of the DNA of that organism, that term we use is genome. Genome. So if you were to say that in each of your cells, you have 23 chromosomes, and those 23 chromosomes altogether are composed of in the vicinity of 3 billion bases, give or take a few million bases or several million bases for that matter. And the 7 million bases encode something on the order of about 21,000 or so genes. That would be your genome. That's your genome. So genomes in bacteria are pretty usually circular molecules of DNA. In eukaryotes, they're usually linear chromosomes. In bacteria, they're usually circles. So they're covalently closed circles. There are exceptions, but that's usually what you see. In cells, genomes are made of DNA exclusively. Viruses can do either DNA or RNA, but in cells, genomes are made of DNA. Mitochondria and chloroplasts also have genomes. Their genomes are also circular. And if you look at the structure of their genomes, they are eerily reminiscent of bacteria. So when we say chromosome, we're talking about a discrete molecule of DNA, linear or circular, that is effectively transmitted to the progeny. So a chromosome essentially, think of it this way. Let's say, and I got this example actually from a student. I thought it was a pretty good example, so I'll give it to you. So her great-great-grandmother had written a letter to her daughter. And she was told not to open it until after she passed. And then her grandmother had written a letter to her mother and was told, don't read this until after I pass. And so she had a letter that was written on the day of her birth by her mother, and it was kept in her thing. She took it with her wherever she went, well, wherever she moved, at least. She didn't bring it to class, but in an envelope that was sealed and had undergone quite a bit of photooxidation from being in light. And she was ready to open it, but not until after her mother passed. And so she said, when I have my first son or daughter, well, my first daughter, it's supposed to be a thing in her family, at least for daughters, but I suppose it could be for sons too. I will write a letter and I will give it to my daughter and I will tell her, don't open it until after I pass. And so the envelope holds a letter The letter has the words. The letter, the envelope, is a chromosome. It's a chromosome. And it contains the information on the letter. And it was passed from great-grandmother to grandmother to mother to daughter. That's a chromosome. We have 23 of those chromatid pairs. We have 23 of those. Each of those is a packet that contains the chromosomes. That's what we mean by chromosome. So in eukaryotes, your chromosomes are linear and they are wound into a structure called chromatin. Chromatin. We won't talk about the structure of chromatin in this class. We will next semester in genetics. For chromatin, you need these proteins called histones. Histones take DNA and they make it into this really compact structure. They wind it into this tight, compact structure called chromatin. Bacteria don't have chromatin, so that's in eukaryotes. For bacterial chromosomes, they have a circular piece of DNA. There can be one or more than one in their genome. And this chromosome in bacteria is wound. It's supercoiled. And that supercoiling will compact the bacterial chromosome into a structure that is nice and compact that can fit inside the cell. So stretches of DNA encode proteins or some sort of RNA that's functional. And we call that stretch that codes for a protein or an RNA. We call that a gene. A gene. Starting with a G, not a J. Now, if a gene is a structural gene, it codes for a protein. And if it codes for an RNA, it's called an RNA gene, frankly. Some genes are regulatory. These regulatory genes may express RNAs involved in gene expression or other things involved in gene expression. So if you take all the genes in your genome and you add them up, that constitutes your so-called genotype, your genotype. Your genotype will then be manifested in the way you look, the traits that cause you to look a certain way. That's your phenotype. Genotype is what your genes look like. Phenotype is what you look like on the outside. So if we pick on Maddie here, for example, Maddie's phenotype is she's tall, she has dark hair, she has brownish eyes. Did I get that right? More or less. She's kind of light complexion, but not super light. I'm guessing you're not Northern European. You're probably more maybe Germanic stock or Eastern European. Okay, yeah, Mediterranean. Okay. Russian. Okay, Fielder's more German. Fielder's your married name. Was your maiden name? Oh, right. Brnovich. Brankovich, which does sound Eastern European. So you're Eastern European just like me. So my guess is in the sun then, you probably tan pretty well since you're Mediterranean. Because us Mediterranean types, we have this trait where we get dark and then in the winter we get pasty white. So we're actually truly colored because we change colors from season to season. So that's her phenotype. That's her phenotype. That's what she looks like. Now, If we wrestled her down and took a blood sample and then sequenced the genome in her white blood cells, that would be her genotype. That's what her genes look like. What she looks like, phenotype. What her genes look like, genotype. Phenotype is a manifestation of genotype. Phenotype depends on genotype. So that they're related, but they're not the same. They're not the same. So in bacteria, you don't have a nucleus. Chromosome is in the cytosol. You might also have these small circles of DNA called plasmids. Plasmids are going to become kind of important in this discussion. They can usually replicate in sync with the bacterial chromosome, or they may replicate autonomously, and you may get multiple copies of these plasmids in some cases. In eukaryotes, your main genome's in the nucleus, but you also have a genome in the mitochondrion. I don't like that picture because the mitochondrial genomes are usually circular. You have a chloroplast genome, and chloroplast genomes are virtually always circular. Viruses, they can do the DNA thing, they can do the RNA thing. Their DNA can be single-stranded or double-stranded. Their RNA can be plus single-stranded, minus single-stranded, or in some rare cases, double-stranded RNA in the case of real viruses. So DNA is a polynucleotide. A polynucleotide meaning it's a polymer of nucleotides. Nucleotides have three parts. They have a phosphate, they have a sugar, and they have a nitrogenous base. A phosphate, a sugar, and a nitrogenous base. In extant nucleotides, the base is a 5-carbon sugar. That's either ribose or a derivative of ribose. 5-carbon sugar, that's either ribose or a derivative of ribose. In DNA, that derivative of ribose is 2-deoxyribose. 2-deoxyribose. You take the hydroxyl group and you get rid of it. You replace it with a hydrogen. That's why it's deoxyribose. The nitrogenous bases, we'll get to those in just a bit, but those attach to the sugar, make sense from the sugar, and the nitrogenous bases tend to not like water. Phosphates are at this pH are charged. They love water. That's going to be important when we talk about the structure of a double helix here in just a bit. So these repeat over and over and over and over and over again. The phosphate attaches to the sugar, that attaches to another phosphate, that attaches to another sugar, that attaches to a phosphate, that attaches to a sugar, and attaches to a phosphate, and over and over and over again. Sugar, phosphate, 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 ad infinitum, ad nauseum, ad negatum secum. That's your polynucleotide, and the bases will extend from the sugar. That's your polynucleotide. Polynucleotides have a front and a back end. These molecules have a polarity to them. So think of them as a train, a choo-choo train, if you will. The engine is the front end, and that front end is called the 5' end. 5' meaning that's the sugar to which the phosphate is attached, and that's the front end. So front and 5 both start with F. That's how you remember that's the front. The front 5' end is the front of the polynucleotide. The tail, the caboose of the train, the tail is the terminal 3' end. They both start with T. The terminal 3' end. That's the tail. The polynucleotides go from front to tail, from front to tail from 5' to 3'. These are highly repetitive molecules. Your bases come in 4 types. There's adenine and thymine and guanine and cytosine. Adenine and guanine are the bigger molecules. They're so-called purines. These are 2-ringed structures. Purines, they're bigger. Pyrimidines are smaller. So adenine and guanine are purines. Thymine and cytosine are pyrimidines. Now, in the double helix of DNA, you have 2 strands of DNA, and these strands of DNA are organized in a very, very particular fashion. The end of this strand here, that's a 3' end. This over here is a 5' end. So this is front and this is tail. When we get to the opposite end over here, so if we follow this one around here, so we said that that over here was the front, this down here, that's the tail. So this goes from front to tail. This other one here goes from tail down to front. That means these 2 strands are in opposite orientation. This one goes front to tail, this one goes tail to front. The phosphate is attached at the front. That's how I can tell easily. And the phosphates are in yellow and red, so they're color-coded. We call this arrangement anti-parallel. You must have these strands in an anti-parallel fashion. If you don't, this molecule does not work. The strands must be arranged in this front to back, back to front sort of arrangement, anti-parallel arrangement. The geometry of the molecule doesn't work if you don't have that. Now, these 2 strands are organized in this fashion with the sugar phosphate on the outside, because the sugar phosphate likes water, and the bases, which don't like water, on the inside. When you do that, to decrease the exposure of the inside to the water, the molecule twists upon itself. To decrease the exposure of the hydrophobic interior and decrease its exposure to water. We call this arrangement the double helix. And these bases on the inside, they are arranged in a particular fashion so that they fit together. Adenine always pairs with thymine. Guanine always pairs with cytosine. This type of arrangement, these bases are called complementary. They're called complementary because in the double helical structure, adenine fits with thymine. Guanine fits with cytosine. This fit means that the diameter of the helix will be constant all the way down. So the diameter of this molecule is about 20 nanometers, and that diameter is the same. So if you look at it from end on, that diameter, it's the same. If you have bases that are mismatched, the geometry of this molecule is going to have a bump in it or a kink in it or something like that. That tells the cell something is not right with DNA because the DNA structure should be pretty constant throughout the molecule. So here is this picture from your book. It shows you the anti-parallel structure. 5' to 3', 3' to 5', anti-parallel with adenine always pairing with thymine, cytosine always pairing with guanine. Notice the adenine and guanine, they have the 2 rings. They're pure rings. The cytosine and thymine, they only have the single ring. They're pyrimidines. So purine always pairs with the pyrimidine. Specifically, adenine always pairs with thymine. Cytosine always pairs with guanine. Now, water is exposed to this. To decrease exposure of the hydrophobic interior to water, you twist upon yourself and you form the double helix. The sugar phosphate backbone's on the outside, the bases are on the inside. And this speaks of the anti-parallel arrangement that I already talked about, so I won't dwell on that. Incidentally, you've probably heard that James Watson and Francis Crick are responsible for solving this structure. Let me correct your historical understanding. They did this by stealing the data of a brilliant X-ray crystallographer named Rosalind Franklin. So it really, instead of being the Watson-Crick structure, it should be the Watson-Crick-Franklin structure. After Rosalind Franklin, and when the Nobel Prize was awarded, she had died from ovarian cancer probably because of exposure to X-rays. The X-ray crystallography machines in those days were not well shielded. And given the fact that she did so much X-ray crystallography, she died of ovarian cancer way too young. Way too young. She was a brilliant researcher, and her death was a loss to science for definite sure. But and then if you ever read J.D. Watson's work, The Race to the Double Helix, he trashes Rosalind Franklin in a way that's very distasteful and also largely untrue. There's this one piece in the book where he talks about she menacingly walked towards me as though to hurt me. Rosalind Franklin was all of 5'2". J.D. Watson was 6'1". Walk toward me menacingly. Right, yeah. Sure thing, James. I actually got to meet James Watson once when I was at UC Irvine. Good gosh, that guy was a jerk. Now, in probably one of the greatest examples of understatement, in their paper in the 1950s on the structure of DNA, they note that it has not escaped us that the structure of DNA lends itself to a method of replication. Namely, if you just unwind the helix, you can use each strand as a template for DNA synthesis. And that presumption turned out to be 100% correct. So this type of replication, where one strand is the former strand, you make a new strand from that former strand. The other strand is a template. You make a new strand and a former strand. So your new molecules will have one new strand and one former strand. That's called semi-conservative replication. I won't go over the experiments that were used to demonstrate that. They're quite brilliant, frankly, but we'll just leave that for a later time. This process is very complicated. You have lots of different enzymes involved, lots of different proteins involved in replicating DNA. So we are going to give you the Reader's Digest version of this. So if you take a look at the handout, you'll see at the top of the handout, there's a picture of what's called a replication fork. Replication fork. Notice that the strands have been unwound and pulled apart. Notice that one of those strands is used as the template for the synthesis of new DNA, and the other is for the synthesis also of new DNA, albeit with a slightly different mechanism. That's your semi-conservative replication. Semi-conservative replication. Your daughter molecule is meant to mimic the parent molecule in its base composition. Mind you, these enzymes do make mistakes, so you will get mistakes usually every so often. Bree, is that some? So the laser beam is, okay, let me do something about the laser beam. You know, I like the sun and everything, but I also like my retina. Better? Okay. And Haley, I see you squinting. If you want that lowered, that's fine. It's okay. You're good. Emily, are you okay with the? You guys kind of get the mule's end with the sun on that end. So if it becomes a problem, feel free to lower that screen there. I won't say a word. Well, probably a word, but not much. So you have one strand is new, one strand is old. That's what we mean by semi-conservative. So let's take our handout and look at some of the enzymes involved in DNA replication. First, you need to unzip the helix, you need to unwind it. That enzyme is called helicase. You can see helicase is that big black dot where the strands are unwound. Helicase takes ATP, it hydrolyzes ATP, it uses the energy in ATP hydrolysis to unwind the DNA molecule. Notice that the single-stranded DNA molecule has these proteins bound to it. These proteins are called SSB, single-stranded DNA protein binding. Single-stranded DNA binding protein, SSB, single-stranded binding protein. SSB binds to the single-stranded DNA and prevents it from reannealing. SSB is not mentioned on here, but you can see it on the handout. Gyrase. Gyrase goes ahead of the helicase because when you unwind the molecule, if the molecule is circular and it's supercoiled, as you unwind it, you put more stress on the molecule, and eventually it takes more and more and more and more energy to unwind the molecule, so much that eventually replication will grind to a halt. So this enzyme called DNA gyrase goes ahead of the helicase and it takes supercoils out of the molecule. By taking supercoils out of the molecule, it now makes life for helicase a lot more easy, makes life easier. Gyrase will be important because that's going to be a target for a very important group of antibiotics called the fluoroquinolones. Gyrase is part of a larger group of enzymes that do this in cells called topoisomerases. Because gyrase is unique to bacteria, it's a target for drugs. Topoisomerase inhibitors are targets for cancer cells. And we will have a look when we get in Pharm to anti-cancer drugs, groups of drugs that attack DNA topoisomerases, of which gyrase is a member of this larger family of drugs. So put that under your pillow and keep it there until next semester. Primase. So for primase, I think a good way to explain primase is to think of track and field. Did any of you, when you were in high school, were any of you sprinters? You're all distance runners, darn. Have any of you been to a track and field and seen the sprinters sprint? What do sprinters start with? Do they start from a dead run, or do they start with something in the what do they put their feet on before they start running? Sprinters blocks, runners blocks, riders block. No, sprinters blocks. So they put their feet on the blocks. What are the blocks used for? So they get a better start. DNA polymerase, which replicates DNA, is a sprinter. It needs starting blocks. It can't start from it's not a distance runner like RNA polymerase. It needs starting blocks. What does it use for starting blocks? It uses a little primer, which is a little stretch of RNA. That little stretch of RNA acts as a starting block for the DNA polymerase. To make that stretch of RNA, you have this enzyme called primase. Primase is an RNA polymerase. Makes a little stretch of RNA, and so the DNA polymerase now has a free 3' end with which to start polymerizing DNA. That's the starting block. The primer here is indicated by the wavy lines. The straight line is DNA, the wavy line is the RNA primer. That primer is made by primase. The main workhorse for DNA replication in bacteria is DNA polymerase III. DNA polymerase III, that's the enzyme that makes new DNA. DNA polymerase I is a repair enzyme that fills in gaps. And there are gaps we'll see that are made during DNA replication. If you have a gap in the backbone, so you have a bond that's missing, you need to repair that bond. To repair bonds in backbones, you use this enzyme called DNA ligase. So it repairs nicks in the backbone. And then the topoisomerases, they're enzymes like DNA gyrase. They go ahead of the helicase and undo supercoils. So those are some of the enzymes involved with DNA replication. This explains it, this shows it, let's go here, and this essentially looks a lot like this. We'll just go through this. So DNA replication on two strands goes in two ways. One of them is continuous, the other is discontinuous. The reason for this is that DNA polymerases, in fact, all polynucleotide polymerases can only polymerize nucleotides in one direction, from front to back. So they make polynucleotides from 5' to 3'. That's the only direction they can go. That's the only direction they can go. So they're like a car that doesn't have reverse. They go one direction and one direction only. So this strand here, where you have the 3' end over here, you make a primer, you start synthesizing, and then you go. This synthesis is continuous. That is the leading strand. Synthesis on the leading strand is continuous. That's why on your handout it says here leading strand. That's continuous synthesis. Over here, you've got a problem because now you have to keep starting, stopping, restarting, stopping, restarting, stopping, so on and so forth. Synthesis over here is discontinuous. This is the lagging strand. Synthesis over here will lag from the leading strand. So primase is going to make these small primers. You'll then add DNA to this until you get to the next bit here and you'll stop. You get up there and then you'll stop. You'll make another primer, you'll go up there, you'll get here, you'll stop. This goes further here, you'll make another primer, you'll go to there and stop, and so on and so forth. These little fragments of DNA with a piece of RNA attached to them Those are called Okazaki fragments. Okazaki fragments. They're called Okazaki fragments after Meiji and Ayako Okazaki at Tokyo University, who discovered them. I saw them give a seminar. I think he was 5'1" and she was 4'10" or something like that. They were tiny, absolutely tiny. But the intellect of these two people far outweighed their physical stature. And for people who lived in Japan all their life, they spoke pretty darn good English too. But trying to locate them at meetings, that was hard. You had to stand up on a chair and try and find them. So these Okazaki fragments need to have the RNA taken out and replaced with DNA. That's what this other enzyme, DNA polymerase I, does. DNA polymerase I is going to chip out the RNA and replace it with DNA. Chips it out, replaces it. What about these gaps here? Those gaps here on the lagging strand will be fixed with DNA ligase. It says here the length of Okazaki fragments are 100 to 1,000 bases long. That's a pretty good range. They can be as short sometimes as 50. 1,000 is a good top end. They can be bigger than that, but that's a pretty good top end. And these are stretches of DNA that have RNA attached at the 5' end. That's the primer. And that's replaced by DNA polymerase I. DNA polymerase I. These enzymes that replicate DNA are very accurate. They make one mistake every billion to 10 billion bases. Extremely accurate. They're not perfect though. They will make mistakes. And then there is the repair machinery that will repair errors made by the polymerases or errors that are introduced by environmental conditions. DNA replication in bacteria is bidirectional. You start at an origin of replication and you extend around the molecule like this. You might get two plasmids that are interlocked. You can resolve them. How do you resolve them like this? That's what those topoisomerases do. They will help resolve these and separate them like that. These molecules too tend to be attached at the membrane. So when the cell divides, one of these will be attached to the membrane over here, the other will be attached over here, so they get divvied up properly to the two different cells. I don't like this concept check question because it's confusing. The way it's worded is confusing. When DNA is replicated, two brand new nucleotide strands are produced. Well, strictly speaking, if DNA replication is semi-conservative, do you get two new strands? Yeah, you get two double-stranded molecules, two new double-stranded molecules, each of which has a single new strand. But is that what it's asking? It's not clear to me if that's what it's asking. So let me ask you this. When DNA is replicated, because replication is semi-conservative, each new double-stranded DNA molecule will have how many new strands? One or two? One. It'll have one, because one strand used for the template, the other strand is new. All right, it is break time. So we'll go ahead and take 5 minutes. We'll reconvene at 1:45, and then we will get back to work with gene expression. You guys are so quiet this morning. There's usually quite a buzz about this place. There's no buzz today. It's a Monday. It is a Monday. Okay, gang, it is time to reconvene. Let's watch this before we go on to our discussion of gene expression.
DNA replication begins when the enzyme replicates unwinds a segment of the DNA and opens up the hydrogen bonding between the two complementary strands of DNA. DNA polymerase, the enzyme that synthesizes DNA, can add new nucleotides only to the free 3' end of a nucleotide strain. To provide a free 3' starting point, a short primer is synthesized first. Synthesis of the other strand of DNA is more complex. To provide a free 3' starting point on this strand, several short primers must be added. DNA polymerase then adds nucleotides to the 3' end of the primers. Finally, the enzyme, DNA ligase, forms a bond between the short DNA strands. During DNA replication, the leading and lagging strands are continuously synthesized.
All right, so that briefly summarizes what we talked about thus far. Go back to PowerPoint here. There we are. So how do you access genetic information within that molecule? You access it by means of making an informational intermediate. That informational intermediate is an RNA molecule. So the making of an RNA molecule from DNA, that process is called transcription. Transcription. So say, for example, your pastor hires you to transcribe his sermons. So you listen to him in church, you sit there with a computer and you click away, but he talks way too fast for you to type. So then you go back to the recording and you listen to it and you type it all up and you try to include the jokes to make sure you got the punchline as well and the dramatic pauses and what have you. And then you've taken the spoken sermon and you've converted it into a written text. That is you transcribing your pastor's sermon. Transcription takes the information in DNA and it remakes it into RNA, a short RNA piece of the DNA. That's transcription. So the main player in transcription is this enzyme called RNA polymerase. RNA polymerase makes RNA, and this is the main player in transcription. If that RNA molecule encodes a protein, that RNA is used to synthesize a protein with a specific amino acid sequence, and that process is called translation. And the main players in translation are ribosomes. Ribosomes. Now, exceptions to this include RNA viruses that have an RNA replicase, Those are enzymes that have a lot of biochemical similarity to DNA polymerase and even RNA polymerases, but they're slightly different. So those are RNA replicases. And drugs like remdesivir and this new drug that in the business section of the Wall Street Journal today, Merck has gone to the FDA to get emergency use approval, molnupiravir for COVID-19. Those inhibit these RNA replicases, at least the RNA replicase in SARS-CoV-2. Retroviruses can convert RNA genomes into DNA copies. That's due to reverse transcriptase. Those would be the exceptions to this pattern. There are also several RNAs that pretty clearly regulate gene function. Those are exceptions to the rule. So the flow of genetic information goes like this. It goes from DNA to RNA to protein. From DNA to RNA to protein. There are cases where RNA can be used to make DNA, like in the case of retroviruses and hepatitis B and cauliflower mosaic virus and things like that. There's no process, at least I'm aware of, where cells can take protein and use it to make an RNA. That's the flow of genetic information. You see that here, where DNA will make messenger RNAs, then these other RNAs, ribosomal RNAs, transfer RNAs, will take part in the translation of messenger RNAs in order to make protein. So once again, from DNA, you make RNA, you make protein, and so on and so forth. Transcription involves a series of steps. The first step involves recognition of a site where the enzyme is supposed to bind and begin transcription. The sequence of DNA, where the RNA polymerase engages the DNA and starts transcription, is called the promoter. The promoter. So it binds to the promoter, it locally unwinds the DNA helix, then it begins transcription, the actual making of the RNA, that's elongation, and then it gets to some sequences that tell it, this is how far you go, that's enough, and that's termination. Initiation is pretty clearly the rate limiting step in transcription. Some genes have promoters that are very good at initiating transcription. They're called strong promoters. Others, they don't do such a good job of initiating transcription. They're called weak promoters. And we'll see in a little bit the difference between a strong and a weak promoter. So RNA is also a polynucleotide. But is chemically distinct from DNA in that instead of 2-deoxyribose for the sugar, you have ribose. The presence of ribose renders this molecule inherently less stable. That's why RNA will be a short-term informational intermediate. Deoxyribose makes the molecule relatively stable. Presence of ribose makes it unstable. Instead of thymine, you have uracil in its place. So DNA contains thymine, RNA contains uracil. Uracil and thymine are chemically very similar. Thymine is actually made from uracil. But RNA will have uracil instead of thymine. Thirdly, when made in the cell, RNA is single-stranded. Bits of the RNA might be able to hybridize with itself. Those are called RNA secondary structures. Those bits that hybridize with itself, they can even form higher order structures. Those are called tertiary structures. Some of those are necessary for function of certain RNAs. So transfer RNAs, for example. The secondary and tertiary structures of transfer RNAs are quite important for what it does. Secondary and tertiary structures for ribosomal RNAs are also quite important because ribosomal proteins recognize ribosomal RNAs by virtue of the fact that they bind to many of these secondary and tertiary structures. But in the cell, RNAs are usually single-stranded. If you get double-stranded RNAs in the cell, they're usually short-term intermediates that exist for regulatory purposes, or if you get double-stranded RNAs in the cell, that usually tells the cell that it's infected with a virus. And in animal cells, if you get double-stranded RNAs, that usually induces the synthesis of this stuff called interferon. Interferon is an alarm bell. When cells secrete interferon, it's an alarm that tells other cells viruses are about to batten down your hatches. And so cells now will start to modify their machinery to make it a lot harder for viruses to enter and co-opt their biosynthetic machinery. That's why interferons, at least recombinant interferons, are used to treat certain viral infections. Are you good with the differences between DNAs and RNAs? So these regulatory RNAs come in 4 types. There are microRNAs. MicroRNAs fine-tune expression. They're seen a lot more often in eukaryotes than they are in prokaryotes. Antisense RNAs, which inhibit translation, you see those in prokaryotes. Riboswitches. Riboswitches regulate genes. When you transcribe the RNA, it'll form this secondary set of secondary structures. The secondary structures will, depending on which secondary structure it forms, will keep the gene turned on or off. That's a riboswitch. You see lots of those in prokaryotes, occasionally in eukaryotes, but more often in prokaryotes. And then SI are small interfering RNAs. Typically, you see those in eukaryotes, not so often in prokaryotes. If an RNA has enzyme activity, which there are a few that do, they're called ribozymes. Ribozymes. These are RNAs that have enzymatic activity. So here's an assembled bacterial ribosome. It's a crystal structure. So if you look at this carefully, you'll notice that you have RNA molecules. Come on, you. You're not wanting to work, are you? Oh, there it goes. So in orange, you have the RNA. And this RNA molecule has these proteins attached to it. These proteins are attaching to tertiary structures in the RNA. So you have a small ribosomal subunit. Right here. That's your small ribosomal subunit. And this up here is your large ribosomal subunit. Large ribosomal subunit. This consists of proteins and ribosomal RNAs. Amino acids are ferried to the ribosome by transfer RNAs. Transfer RNAs have this interesting cloverleaf type secondary structure. The end of the transfer RNA always ends in C, C, A at the end. C, C, A, and then this is the 3' end right here. Three' end like that. The CCA is added after transcription. So you make your tRNA, it doesn't have the CCA at the end. Then you tack on the CCA after transcription. All transfer RNAs end in CCA. Then they fold into these 3 loops like this. So one of these loops essentially has this thymine right here. Oh, wait a minute. RNAs don't have thymine. This one does. That thymine is introduced after transcription. That's a post-transcriptional modification of the tRNA. Because of this, this is often called the thymine loop. Over here, you have another loop. And then this loop here is the anticodon loop. The anticodon is going to interact with the messenger RNA on the ribosome directly. Now this secondary structure folds into this very sophisticated tertiary structure. So these 2 loops here are going to clap their hands kind of like that. This is going to then go this way. And then this particular anticodon loop heads straight down. So the whole molecule now will do this, this, and this. So the molecule has an L shape to it. And these 2 loops here, the thymine loop and this loop down here, they are hydrogen bonding together like this. They're kind of clapping their hands like that. This then bends this way, and this bends down that way. This is the tertiary structure of your tRNA. Your amino acid gets attached right here, and this anticodon interacts with the messenger RNA. Your messenger RNA, the ribosome will read it in groups of 3 bases. We call those groups of 3 bases codons. It's hard to write using the mouse. I don't do that very well. I hate it when you have to sign on screens like that. I hate that. I can't do it. I get something that looks nothing like my actual signature. So these codons, they're going to essentially be able to hybridize with the anticodon. Now this goes from, this is red, from 5' to 3'. This, therefore, is going to go in the opposite direction. So this is going to go from 5' this way to 3' that way. And if the anticodon is complementary to this codon here, that will be the right tRNA for the codon here. Now, your ribosomes between eukaryotes and prokaryotes have a similar structure, but they're different. They're different sizes. Ribosomes in bacteria, ribosomes in mitochondria, and chloroplasts, yes, mitochondria and chloroplasts have ribosomes. The ribosomes in mitochondria and chloroplasts, again, are very eerily bacterial looking. They are so-called 70S, 70S. S stands for Svedberg. Svedberg, what's a Svedberg? Svedberg is how fast you descend in a centrifugal field. There's two things that influence how fast you descend in a centrifugal field: size and shape. Size and shape. There's two subunits to the prokaryotic ribosome. A small ribosomal subunit that's 30S, and a large ribosomal subunit that's 50S. Now, I know 50 and 30 don't add up to 70. The reason for that is that once you get to putting these two together, that changes their size, their shape. Because it changes their shape, that modifies their descent in the centrifugal field. In eukaryotes, you have 80S ribosomes composed of two subunits, a small 40S subunit and a large 60S subunit. Yes, I know 40 and 60 don't add up to 80, but again, you assemble it, you now change the shape. And because you change the shape, that changes the way it descends in the centrifugal field. So the small subunit initially binds to the messenger RNA first. It binds to the 5' end. Translation always begins with the codon AUG. Always. AUG codes for the amino acid methionine. So all proteins will have as their first amino acid, at least when they're initially made, methionine. And why don't all proteins have methionine at the amino terminus? Because there's an enzyme that will actually clip the methionine off. But plenty of proteins have methionine at the beginning. So there's 3 sites on the ribosome: A site, P site, E site. You can remember this by remembering APE. APE. So the methionine tRNA, which is the starting tRNA, it's going to bind, and the AUG will be here. You have an anticodon here that is complementary to the AUG. And then the very next codon is found at the A site. When the proper tRNA that is carrying the amino acid to which it corresponds occupies this site, you now activate in the ribosome something called the peptidyl transferase activity. Peptidyl transferase activity. You make a peptide bond between these two amino acids. And this one gets transferred over here. And then the ribosome notes that it has an empty tRNA here. So what does it do? It hydrolyzes the equivalent of ATP and it moves over like this. And this one moves over here, and now this one's empty. This goes to the E site, the empty site, and now its affinity for the ribosome is low. So this tRNA leaves, it gets charged up with another amino acid, it collects go, passes go, collects $200, and does the whole thing all over again. When these two sites are occupied, the A and the P site are occupied, and you activate the peptidyl transferase activity in the ribosome, the peptidyl transferase activity of the ribosome maps to the large ribosomal RNA. The peptidyl transferase activity is due to a ribozyme in the ribosome. So all the proteins in your body are actually made by a ribozyme. So which codons correspond to which amino acids? That is the stuff of the so-called genetic code. The genetic code, which we can see right here. Because you have 3 different bases coding for 20 different amino acids, that means there's 64 possible different combinations. You'd expect there to be some redundancy, and there is. So something like arginine is coded for by 6 different amino acids. Something like leucine is coded for by 6 different amino acids. Methionine is just coded for by 1 amino acid. Others are coded for by 2 or something like that. There are 3 codons that don't code for any amino acid. All of those are stop codons. So they all start with U. There's UAA, UAG, and UGA are the 3 stop codons. When the ribosome gets to the stop codon, no tRNA binds. So the ribosome says, okay, that's enough. We've made our protein. And instead of the peptidyl transferase transferring an amino acid to another growing polypeptide chain, instead, it just clips the protein from the tRNA, and the protein is now free. So that's the genetic code. Now, if we look at the genetic code, we'll notice that the difference in, say, these codons here, the first and second positions are the same, but the third position is the one that differs. The reason for that has to do with the fact that your tRNA when it interacts with the bases here, that number 3 position, the base is the furthest away. Because the base is the furthest away, the pairing rules get relaxed. Those relaxed pairing rules is what we call wobble. Wobble. W-O-B-B-L-E, wobble. And this permits variation in the codon without altering the amino acid sequence that the messenger RNA codes for. So you could theoretically look at a sequence and know exactly what protein it's going to make, and that's essentially what you can do. Okay, we don't have class on Friday, but then a week from today, we will clean up and talk more about translation. Translation is a major target for antibiotics. So we'll take a look essentially at some of the stages because different antibiotics are going to hit different stages. Some will overlap. And then we'll look at essentially control of gene expression. That gets a little complicated. That gets a little hairy. So we'll use videos and I'll try to bring in my best analogies and what have you. Tomorrow, we're going to continue our biochemical experiments. We'll do IMVIC tests. IMVIC tests are extremely important for GI and urinary tract infection types of organisms. And the labs we did last week will be due, but not 18. So all the labs we did, but not 18 will be due. Okay? All right, Natalie, Bree. Good to see you guys. I hope you guys are.
Nice to see you too. Sorry, my alarm never went off. So, yeah. Like my phone never charged because I used a new charger and then my alarm never ended up going off.
Okay. I have your quiz. I'll just give it to Mrs. Schaefer. You can pick it up from Mrs. Schaefer. You already have your lab book, right?
Yes.
All right. So Mrs. Schaefer will have your quiz. Oh, you know what? Scratch that. I'll just bring your quiz to lab on Wednesday, unless you want it earlier, and I can. If you want it earlier, I can give it to Mrs. Schaefer. You can get it from Mrs. Schaefer. That's up to you.
Okay, Wednesday will work perfect for me.
Okay, I'll just bring it to lab.
Perfect. Thank you very much.
And I'll see you Wednesday.
See ya.
All right. Ciao.